Last time we have stopped here. Now we will see about the transportation through cell membrane. So how transportation occur from one part of the membrane to the other. So this is we will see in detail. What needs to cross plasma membrane or cell membrane? So nutrients, wastes, molecules, fluid, and certain ions. Plasma membrane is a very important structure which functions to allow certain substances to enter or leave the cell, still excluding others to cross or to pass cell membrane. Cell membrane or plasma membrane can pump other substance into and out of the cell against the concentration gradient. So this such membrane is called an selective permeable membrane or semi-permeable membrane. Selective means it passes by selection from the other. So it's semi means by half. It's the same meaning. So it is cell membrane is selectively permeable or semi-permeable. Both protein and phospholipid portion of the membrane are involved in the membrane permeability. In this selective permeability, both protein and lipids are participate in this way. Transport through cell membrane. By the way, we do have two type of categories. The one is membrane used for passing ions or small solid molecules. The other is large molecules. So the large molecules we can say vesicular transport, that includes like endocytosis and exocytosis. So but membrane, there are two categories of transport: the movement of ions or small molecules to pass through cell membrane. The one is passive transport, the other is active transport. So we do have so this is we do have two categories of transport. The one is passive transport, the other is active transport. This is for movement of small solids or ions. So transportation for ions or small molecules is can be classified into passive transport and active transport. So in passive transport, transport of substances without use of energy. So in case of passive transportation, cells or the transportation process didn't use or does not use the energy. It is energy free. So this passive transport includes simple diffusion, facilitated diffusion, and osmosis. These three things are free from energy. That does not use energy. So under passive transport we will see simple diffusion, facilitated diffusion, osmosis. The second type of category is active transport. Active transport use energy or ATP from cells reserves to power transport of substances to get energy. So active transport can be divided into primary active transport and secondary active transport. The other type of transportation we do have the other type of transportation is transportation of large molecules or large particles. These large particles are transported via vesicle-like sac across cell membrane. The process of passing large particles through cell membrane via vesicle is called vesicular transport. So vesicular transport divided into two: endocytosis and exocytosis. These all types of transportation we will see one by one. So from passive transport, now passive transport can be classified into three. The first is simple diffusion, the second is facilitated diffusion, the third is osmosis. So now we are going to simple diffusion. Diffusion is a passive movement of substance down their concentration gradient. So passive movement means it does not use energy. So by the way, simple diffusion is a movement of substances down their concentration gradient, means a movement of substances from higher concentration to the lower concentration. So factors affecting the net rate of diffusion is electrical potential difference of ions, one, the lipid solubility of substances, concentration difference or pressure difference, and permeability, membrane permeability is factors affecting the net rate of diffusion. The rate of diffusion is affected by lipid solubility, concentration difference or pressure difference, and membrane permeability. And membrane permeability is affected also by the thickness of the membrane, thick membrane and thin membrane. The number of ion channels per unit area, how many channels are found in unit area, affects the permeability. The other is temperature. As temperature increases, thermal motion of molecule increases, so the permeability increases. So temperature affects permeability. The other is molecular weight. Molecular weight is also affected permeability. So the rate of diffusion is determined by the following factors summarized in the formula. So rate of diffusion is equal to CA/Ts over D under square root of molecular weight. So C is change of concentration. S is this S is solubility in lipid of substances. A is surface area of the membrane, whereas T is temperature. D is membrane thickness or distance. The other MW is molecular weight of substance. So example, substances that are transported by simple diffusion are an example of simple diffusion substances. An example of substances that are passing in simple diffusion are carbon dioxide, oxygen, alcohol, lipid soluble drugs, and ions that are passing through specific channels. So the second type of passive diffusion is facilitated diffusion. As the name indicates, facilitated, it is more rapid than simple diffusion. So as it is simple diffusion, it does not need energy and transports substance down their concentration. Transport substance down their concentration gradient. So facilitated diffusion is transport substance down their concentration gradient. That means a movement of substances from higher concentration to the lower concentration. It is more rapid than simple diffusion. And the very important thing is facilitated diffusion is carrier mediated. It's carrier mediated. So therefore, it exhibits zero specificity, saturation, and computation. So carrier mediated means they use carrier protein for transportation. So this is make it different from the simple diffusion. An example is transport of glucose and protein. That's macromolecule. So transport of glucose and protein is known as facilitated diffusion. So carriers are okay. Let us see. This is extracellular fluid. This is membrane, cell membrane. This is intracellular fluid. So this is carrier protein. So this is glucose here. The glucose is going from the extracellular fluid to the intracellular, from higher concentration to the lower concentration by carrier mediated protein or by using carrier proteins. So this is an example of facilitated diffusion. The facilitated diffusion carriers are saturable. So fluid flux increases with extracellular concentration only up to certain point. There is limitation. So unlike simple diffusion, the rate of facilitated diffusion increases as the concentration gradient increases until all the carrier sites are filled. That means in simple diffusion, as concentration gradient increases, the diffusion also increases. There is no limitation. But in the case of facilitated diffusion, as concentration gradient increases, the diffusion also increases, but up to certain limit until carrier sites are filled. As carrier sites are filled, it will be uniform. So at this point, the rate of diffusion can no longer increases with increasing particle concentration. This point, this stage is called saturation. Saturation means as the concentration gradient increases, there is no longer increasing in particle diffusion. As concentration gradient increases, there is no longer increasing particle concentration. So this is called saturation. So let me show you. This is the rate of diffusion. This is the concentration of substances. As concentration of substances or concentration as concentration difference increases, the rate of diffusion increases. So in the case of simple diffusion, as concentration of substance increases, the rate of diffusion increases. There is no limitation. This is simple diffusion, so there is no limitation. As this increases, it will increase. So simple diffusion is this. But in the case of facilitated diffusion, as concentration difference increases, the rate of diffusion increases up to certain limit, Vmax, up to certain limitation, meaning until carrier sites are filled. So after that, there is no increase in diffusion, so it will be uniform. This is known as facilitated diffusion. So this is for facilitated diffusion. This is for simple diffusion. So the third type of passive transport is known as osmosis. So osmosis is a movement of water from an area of higher amount of water to an area of lower amount of water through semi-permeable membrane or through selectively permeable membrane. That means also the same from higher concentration to the lower concentration, but this is for limited to water. So in the case of diffusion, they are molecules, but in the case of water, in the case of osmosis, it is water. Water moves from higher concentration of water to the lower or higher amount of water to the lower amount of water across the cell membrane. So the direction of movement of water is governed by or depend on the amount of osmotically active particles or solutes. That means this is, for instance, this green one is sugars, so this is solutes. So the amount of osmotically active particles means it is the amount of these solutes. The direction of the movement of water increases. That means there is no solute here, there is no solute here, there is no solute here, there is solute here. So the direction of the water is from this part, from the left to the right. Why? Because the amount of osmotically active particles found on this side, the left side. So particles are found or solutes are found on the left side. So the movement of the water is from the right to the left side, on the anatomical position. So when we see this osmosis, this color is water, this color is sugar. So this is the membrane, selectively membrane. This is for instance, if you say this is one compartment of the cell, is the compartment. So in this case there is only water. In this case there is water plus solutes. These solutes are sugar. So this is low sugar concentration, this is high sugar concentration. So if there is solutes or sugar, there is low water concentration. In this case, high water concentration is found. So by this case, the movement of water from this is high concentration, this is low concentration. Water is moved from the high concentration to the low concentration, from the left to the right. to the right to left in anatomical position. So this is right, this is left, and water is moved in this direction. So this is an important example of osmosis. So osmosis is depend on two things. The one is difference in solute on those types of plasma membrane. The other is membrane must be impermeable to the solute. These two things are very important requirements. I will show you. Osmotically active solutes means solutes that cannot pass freely through membrane. So membrane must be impermeable to solute. So difference in solute into sides of plasma membrane. One, to osmosis to be occur, one solute difference should be occur. So this is high solute area, this is low solute area. So in order to osmosis occur from low solute to high solute, in order to osmosis occur from high concentration water to the low, should solute difference should be present. High solute, low concentration water, low solute, high concentration water. So solute amount is important for initiation of osmosis. The other is this cell membrane is impermeable to these solutes, otherwise solutes will pass just like this. So this membrane is impermeable, so waters are going to from less soluble to high, less solute area to high solute area, or if there is less solute, it's high amount of water. So from high amount of water to less amount of water will be the direction of the water or osmosis. So the two things are very important. So osmotically active means impermeable or cannot pass through this cell membrane. So they are important for initiation of osmosis. So these two things are very important. So the other type is type of category is active transport. So we have completed passive transport, that is diffusion, simple diffusion, faster diffusion, and osmosis. There is active transport. Category is active transport. Active transport, in the active transport, substances are transported against concentration gradient uphill direction, meaning from the lower concentration to the higher concentration. That means against concentration gradient or uphill direction. This is consumes energy in the form of ATP, meaning this is uses active transport use energy, passive is does not use energy. So carrier protein is involved. So like active transport, faster diffusion we also use carrier protein, but simple diffusion does not use carrier protein. So ions or elements used in the transportation of active transport is sodium, potassium, calcium, iron, hydrogen, and chlorine are an examples of active transport. So when we see active transport can be divided into two, primary and secondary active transport. So the primary active transport, an example for primary active transport is sodium, potassium, ATPase. Sodium, potassium, ATPase. That means sodium and potassium ATPase is an example of primary active transport. Calcium ATPase pumps calcium into sarcoplasmic reticulum or plasma membrane. The other is hydrogen potassium ATPase, or we can say proton pump. These three things are an example of primary active transport. So in this case gastric parental cells transport hydrogen into the lumen of the stomach and maintains low pH in the stomach. Low pH means acidic. So it is this acidic environment is inhibited by omeprazole. So from the process of hydrogen transport into lumen is an example of hydrogen ATPase or hydrogen potassium ATPase or proton pump. So before that, let me tell you some important terms about what is osmolarity, or something like that. I will come back, but let me tell you about osmosis. In the osmosis, the osmosis, there are, let me tell you, some osmolality and osmolarity and osmolality are frequently confused and words interchangeably used. So osmolarity refers to osmotic pressure generated by dissolved solute molecules in 1 liter of solvent, whereas osmolality is the number of molecules dissolved in 1 kilogram of solvent. So the difference is here. So osmolality is a pressure generated by dissolved solutes in molecule in 1 liter of solvent, whereas osmolality is the number of molecules dissolved in 1 kilogram of solvent. So the second difference is also osmolarity is 1 liter in 1 solvent, whereas osmolality is 1 kilogram in solvent. So this is a difference. So for dilute solutions, the difference between osmolarity and osmolality is insignificant. Measurements of osmolarity are dependent on temperature because volume of solvent varies with temperature. For instance, if the volume is larger when temperature increases, where there is higher temperature. So in contrast, osmolality is based on mass of the solvent, so it is independent of temperature, meaning osmolarity is dependent on temperature, whereas osmolality does not depend on temperature, but osmolality is dependent on mass of solvent. So osmolarity is expressed later in 1 liter, whereas osmolality is expressed in 1 kilogram. So this is a difference. So the other important thing is what is osmosis? Osmosis is the power of solution to dry the solvent through semi-permeable membrane. Osmotic pressure is the pressure needed to prevent the movement of solvent across semi-permeable membrane into the solution. And the other term is osmosis. Osmosis is the ability of solubles to cause osmosis and osmotic pressure. So measured in osmoles, 1 osmole is a gram substance of the molecule dividing by the number of freely moving particles on solution. Osmolarity is the concentration of osmotically active particles per liter of solution, osmole per liter. That means 1 osmole of the solute added to 1 liter of solvent. Osmolality is a concentration of osmotically active particles per kilogram of solution. That means osmoles per kilogram. 1 osmole of solute and 1 kilogram of solvent are added together. This is an important term about osmosis. So now let us pass about active transport. Active transport have primary active transport, secondary. Primaries are sodium, potassium, calcium and hydrogen, potassium. ATPase is an example of primary active transport. So transportation of hydrogen to the lumen of stomach and maintains low pH acidic environment in the stomach is an example for primary active transport. Whereas let us an example of, so we have seen hydrogen, we will see calcium, and now we are ready to observe sodium, potassium, ATPase. Active transport, that is sodium, potassium, ATPase, primary. So sodium potassium pump is a carrier protein that is made up of 2 subunits, 3 binding sites for sodium inside the cell, 2 binding sites for potassium outside the cell. Outside cells there is 2 binding sites for potassium. Inside the cell there is 3 binding sites for sodium. So it pumps 3 sodium outward and 2 potassium inward. It maintains electropositive outside, electronegative inside. Both sodium and potassium are transported against their electrochemical gradients. It has ATPase activity inside the cell, so ATP divide into ADP plus phosphate, so it forms energy. It's inside the cell. So energy brings conformational change of the pump, so that sodium pumped outward, potassium pumped inward. So creates an electrical potential across cell membrane. So now let us see an example. This is cell membrane. This is a channel, sodium potassium pump channel. So in this case, as this area, ATPs will break down into ADP and phosphorus. So this produce energy inside the cell. This energy, this is ATPs channel. So this energy makes conformational change of the to pump sodium to outside to and potassium inside. So the sodium, 3 sodium inside will pass into outside the cell, from inside the cell to the outside cell. So inside 3 sodium will move into outside into 3 sodium, whereas potassium outside will move into inside the cell. So this energy will make conformational change and will go into concentration gradient, will go into will transport it into against concentration gradient. That's why, because sodium are, by the way, sodium are too much in extracellular fluid and less in intracellular fluid, whereas potassiums are high in intracellular fluid and low in extracellular fluid. So sodium moves from inside to outside, so it's against concentration gradient because it moves from the lower concentration inside the cell to the higher concentration outside the cell, whereas potassium is also against concentration gradient because since potassium is high in intracellular, potassium is moved from outside to inside, means against gradient from the lower concentration to the higher concentration. So this is an important concept of sodium potassium pump. So ATP energy formation is occur in intracellular fluid or on the inner side of the cell membrane, whereas it's outside, so will lead to the direction of sodium and potassium. So this is an important part of sodium potassium ATPase. It is primary transport, active transport. So another example of is potassium. For instance, substances are transported against concentration, electrochemical gradient, uphill direction. So calcium pump or calcium ATPase are found in the plasma membrane, in the membrane of endoplasmic reticulum, or in muscles in the sarcoplasmic reticulum. So there are also P-type of ATPase. P-type means it's proton type ATPase. They pump calcium ion from cytosol of the cell, either into extracellular space or into the lumen of these organelles. Organelles store calcium as a result help to maintain low cytosolic concentration of this ion. So this is an example for calcium ATPase. But hydrogen potassium ATPase, as we have seen, it's another example of P-type ATPase or proton type ATPase. It's present in the luminal membrane of the parental cells, in oxyntic or acidic secreting glands of the stomach. So by pumping protons into the lumen of the stomach in exchange for potassium ion, this pump maintains the low pH in the stomach that is necessary for proper digestion. It's also found in the colon and in the collecting duct of the kidney. Its role of the kidney is to secrete hydrogen ion into the urine when blood pH falls and to reabsorb potassium ions. And another example is proton pump or hydrogen ATPase. ATPase are found in the membranes of lysosomes and Golgi apparatus. They pump proton from cytosol into the organelles, keeping the inside of the organelles more acidic, low pH than the rest of the cell. So they are also found in the plasma membrane of bone and kidney cells. The secretion of protons by bone cells, that's osteoclasts, helps to solubilize the bone mineral and create an acidic environment for the bone breakdown by enzymes. So the proton pump in the kidneys present same cells intercalated as hydrogen potassium ATPase and helps to secrete hydrogen ion into the urine when blood pH falls. So now we have discussed sodium potassium ATPase, an example of sodium potassium ATPase, calcium ATPase, hydrogen and potassium ATPase, and hydrogen ATPase. So this is what we have seen is an example of these three things. Now secondary active transport. Secondary active transport is as the primary, its carrier protein is involved and consume energy. It makes these two things makes it similar with primary active transport. The movement of molecule down its concentration gradient, usually sodium, provides energy for the uphill transport of the other solute. Metabolic energy is not directly used but indirectly from sodium gradient. This means this is down to concentration, from high to the low concentration like sodium, but for the other molecule it's uphill that uses energy, but energy is indirectly. Let me show you. This is membrane, this is a channel, the channel. So in this case this is sodium binding site, and this is sodium, and this is glucose. Sodium is moved from the outside to the inside, meaning against meaning molecule down its concentration from high concentration to the lower concentration. But for glucose it is uphill against concentration gradient from outside to the inside. So this is glucose binding site. So meaning sodium is from outside from high concentration to the low concentration. So mostly in the secondary active transport sodium is present. So when sodium moves from outside to inside, it makes it to start movement of the glucose. So the glucose uses energy. That means that's why we used indirectly energy. It's not for sodium but for glucose. So this type of transportation is secondary active transport. So sodium is bind with glucose. Since sodium moves from the higher to the lower, it makes to initiate the glucose movement from low to higher concentration. So in that case energy will be used as indirectly. So this is a very important thing. So so the bindings of more than one molecule in one direction is symport or co-transport. So sodium plus glucose co-transport in the small intestine, sodium, potassium and chlorine co-transport in the kidney. So meaning sodium is present. So glucose is bind with sodium to enter the cell, and potassium chlorine is bind with this type of transport is can be named co-transport or co-transport or symport. So all co-transports have sodium as one of the transported solutes except in the yeast. That means all solutes, all transports have sodium. So one or more than one solute with sodium is binded in the case of transportation. So this is an important example. So energy is used for the solute. In this case, the other is depending on the number of substances that carriers transport. Carrier protein may be referred to as the carrier protein is referred to as uniport carriers, antiport carriers, and symport carriers. Uniport carriers carry a single substance to one direction. With one direction, carry a single substance is called uniport carriers, whereas antiport carriers carry two substances in opposite direction, facilitating exchange of substances. Symport carriers carry two substances into the same direction is symport carriers. For instance, symport carriers is here is symport carriers because sodium and glucose is go into two substances goes into the same direction is symport carriers. So we do have three types of carriers: uniport, aniport, and symport carriers. Symport carriers, and so this is an important thing about carriers. So transport through cell membrane as revision. This is simple diffusion. Simple diffusion is used channel. This is channel protein. So simple diffusion is across the cell membrane, either inside to or outside. So this is exchange of materials. This is called simple diffusion, either from high concentration to the lower concentration of molecules, either inside to outside or outside to inside. But in the case facilitated diffusion, this is known as not channel protein, but it's carrier protein. It's carrier protein, so it's extracellular fluid, this intracellular fluid use carrier protein. So if use carrier protein, we can say facilitated diffusion. Facilitated diffusion is more faster than simple diffusion. Simple diffusion, so the other type is, this is also carrier protein, this carrier protein, but uses energy. This is called active transport. Active transport is active transport, so this is active transport. Carrier protein plus energy is active transport. Carrier protein without energy is facilitated diffusion. And simple diffusion also does not use energy. So this is an example of transportations. So another additional example is to show, these examples are to show passive, facilitated and simple diffusion, facilitated diffusion and active transport. In passive diffusion or simple diffusion, this is down concentration gradient. This means high concentration to the lower concentration. So this is friction here. So movement of this material from high to the lower concentration. But this is facilitated diffusion, similar with passive diffusion. The difference is this is half friction, this is less friction because it uses tires. So this is faster than from this one. So facilitated and passive diffusion, they are they are similar. So the movement is from high concentration to the lower concentration. So it is down concentration gradient. So the difference is their speed. Because of this. The other is active transport. Active transport are uphill against concentration gradient from the lower to higher concentration gradient, but it uses tires, so it's very strong heaviness, uses energy because of these cars are used. Since these cars are used fuel or gases. This one didn't use fuel or gas. This is uses, since its car uses gases. A similar active transport uses ATP or energy. So this is an important example for serial transportations. The last type of transportation is vesicular transport. This transport is for macromolecules or large particles or molecules. Vesicular transport can be divided into two. The one is endocytosis. The other is exocytosis. So endocytosis means from outside to inside, whereas exocytosis means from inside to outside. From endocytosis, cells internalize extracellular material, meaning cells engulfs outside material, engulfing materials by invaginating the outer parts of the cell membrane until it buds off within the cytoplasm, until it's joined into cytoplasm. By the way, so this is outside material, so engulfs until this material is goes past these steps and internalized into cytoplasm. Endocytosis will occur. Endocytosis can be divided into two. The one is phagocytosis or cell eating. The other is pinocytosis or cell drinking. So when we see phagocytosis, cell eating is a process by which bacteria, dead tissue, other material are engulfed by cells. Engulfed means swallowed by cells. Substance is solid. The difference is, in case of phagocytosis, the substance is solid, whereas in the case of pinocytosis, the substance is liquid. So phagocytotic cell or macrophages are almost equal size to engulfed. In the case of pinocytosis, they are similar in size, but in case of phagocytosis, macrophages or the cell and the engulfed substance is equal in size. So this is material. So this material is in contact with cell membrane. So the cell membrane is folded and cell membrane are fused together, and this material is engulfed and form vesicle here. So this is known as the process of internalized external material or engulfing from outside to inside or cytoplasm is called this process is called endocytosis. But if this material is so solid, we can say phagocytosis, but if this material is liquid, we can say pinocytosis. So pinocytosis, cell drinking, similar process like phagocytosis, but the substance ingested are solution or liquid. Vesicles are much smaller in size as compared to the cell. The vesicles are smaller in size, but in the case of phagocytosis, they are equal in size. Vesicle and the cell are equal in size. So invagination occurs into the cell and pinches off to form boundary of an extracellular vesicle, vacuole or tubule. So as you see in the picture, here is the material. So if it is in contact with cell membrane, the cell membrane initiates invagination, and by this time, so it will change into seems like this phagocytic vacuole, then will change to phagocytic lysosome, then finally it will form just like this. So the outside fluid is drunk, so form just like this. So this is known as pinocytosis or cell drinking. So pinocytosis plus phagocytosis is endocytosis. That means anything outside the cell will be entered into the cell. But in the case of the second type of vesicular transport is exocytosis. This is known as cell vomiting. Vomiting means will remove out inside cell contents. So in the case of exocytosis, neurotransmitter, digestive enzymes and some hormones are released from inside the cell to the outside the cell. So this enzyme or neurotransmitter or hormones are found in the cytoplasm. Then they are found in secretory vesicle. So this vesicle in contact with the cell membrane. So the cell membrane forms just like this and will vomit or remove out these hormones or enzymes, hormones and enzymes. So vesicular transport can be divided to endocytosis and exocytosis. So the difference is vesicles or other bodies in the cytoplasm move macromolecules or large particles across the plasma membrane. So exocytosis means describes the process of vesicles fusing with plasma membrane and releasing their contents to the outside of the cell. This process is common when cell produces substance for export. So when cells are producing, for instance, protein or any substance, if the cell wants to remove some substance from inside the cell to outside the cell, it uses exocytosis. But in the case of endocytosis, the capture of substance outside cell when plasma membrane merges to engulf it. So the plasma membrane merges with external cell and it engulf it or swallow it. So the substance subsequently enters the cytoplasm, enclosed in vesicle. So external material are enclosed in vesicle. So if it is in contact with cell membrane, the cell membrane engulf it and will join into the cytoplasm. So if the cell wants to enter some substance into the cell, it uses endocytosis. For solid is phagocytosis, for liquid or solution is pinocytosis. So this is all about chapter 2, that is cell and membrane physiology. Thank you very much for your attention. Have a nice time. To get the next videos and different courses and also this physiology course, don't forget to subscribe this channel. Have a nice time. Bye bye.